Now look at this.
What's up, penguins? Today we're going to do topic 2.2 on cell structure and function. So the RefER is going to be studied with ribosomes. What this means is that there's ribosomes that are on the membrane of the RefER. The proteins are going to be synthesized by the ribosomes. So since they're sitting on a membrane, we can make membrane proteins, or proteins in the membrane, or proteins that are surrounded by membrane. So this would be for secretion, as well as it can make a couple of our different organelles. So what happens is after it's formed, it goes into a little vesicle that then moves over to the Golgi where it packages and sorts it to move it throughout the cell or to allow it to go through exocytosis to leave the cell for secretion of those proteins. Lysosome is an example of one of the organelles that can be formed due to the RefER. So the RefER is going to synthesize the hydrolytic enzymes or the enzymes that are responsible for hydrolysis that will then be stored within the lysosome. The lysosome is going to help to digest or break down some of these macromolecules that may be non-functional. So if you have a non-functional protein, it can be broken down by the lysosome as well as there's a non-functional organelle. So this leads us into apoptosis. Apoptosis is where we have programmed cell death. So it gets some type of death signal. That death signal then causes it to have the nucleases and the proteases start breaking down the nucleic acids and the proteins, which then allows it to kind of break down from the inside out, which is what happens in apoptosis. So here's our mitochondria. So a lot of the stuff we need to know for the mitochondria comes into play in Unit 3. But we do need to know about where certain things are taking place in the mitochondria. So number one, we see that we have this inner membrane. That inner membrane is called the cristae. The cristae is where we're going to see oxidative phosphorylation. And part of oxidative phosphorylation is something called an electron transport chain. An electron is going to drop in energy, and as it does that, it pumps protons across the membrane. The protons move into this inner membrane space, so the space between the inner membrane and the outer membrane. And then we also see that in the matrix, on the inside of that cristae, the matrix, that is where we're going to see the Krebs cycle taking place. It's important to note that both plant cells and animal cells have this organelle. And so don't get tripped up when they ask you about where does electron transport chain take place. It will take place in the mitochondria as well as the chloroplast that we'll see in just a moment. So here's our chloroplast. As we said before, this is where photosynthesis takes place. So you see that it has those little stacks. We said before that that's called the thylakoid. Inside the thylakoid membrane, you have a pigment called chlorophyll. Chlorophyll is going to absorb the light energy. And so in the thylakoid membrane is where we see our light reactions taking place, which we'll see another electron transport chain, again, pumping protons across the membrane into the thylakoid space to generate a proton gradient that will allow us to make ATP. Inside the stroma of the chloroplast is where we're going to see the light-independent reactions, aka the Calvin cycle, take place. Remember, I'm doing this for all the units, so hit that follow button. And remember, AP Bio Penguins, dress success. Bye, y'all.